Mahur, Assistant Professor, Zoology, Sri Samlaji Government College, Mandafia, Chittorgarh. Today my topic is models of plasma membrane. Every living cell is extremely covered by a thin, transparent, electron microscopic, elastic, regenerative, and semi-permeable membrane, which is called plasma membrane. It is also known as cell membrane or plasma lemma. It is a network of lipids and proteins, and that forms the boundary between a cell contents and the outside of the cell. Its peculiar functional property is its semi-permeable nature, and it regulates the material that enter or exit the cell. It is primarily meant to regulate the exchange of material between the cytoplasm and extracellular environment. Hence, it maintains homeostasis inside the cell. And additionally, it also helps in synthesis of many macromolecules, response of the cells to a variety of physical and chemical stimuli. So, main function of plasma membrane is to protect the cell from its surrounding environment. To understand the structure of plasma membrane, there are two types of model, that is laminar model and fluid mosaic model. Lamellar model of plasma membrane includes Danieli and Davison model, and second it's Robertson's model. So first of all, I will explain Danieli and Davison model. This model is also known as sandwich model. It is also known as protein lipid protein sandwich. It was proposed by J. Danieli and H. Davison in 1935 AD. According to this model, plasma membrane is lipoproteinous and trilamellar or tripartite membrane. It is formed of outer and inner protein monolayer and middle transparent phospholipid layer. Hence, this model is known as protein lipid protein sandwich. So, protein, lipid, and protein. Protein molecules are globular, that is alpha protein, and it provides stability to the plasma membrane. Now, phospholipid bilayer. In this phospholipid bilayer, there are phospholipid molecules. See, these are. Each phospholipid molecule is a polar molecule and it has two specific ends. First is hydrophilic, that is polar or glycerol or head, and hydrophobic, non-polar or fatty acid or tail end. So each phospholipid molecule is an amphipathic molecule. Heads of phospholipid molecule these are directed in opposite direction and are associated with the protein molecule by hydrogen bond, ionic linkage, and electrostatic force of attraction, while the tails of two layers face each other. Each plasma membrane is a porous membrane and they have microscopic pores of 7 to 10 angstrom. This model has some drawbacks. First, it doesn't explain the functional specificity and variability in the biomembrane. According to this model, plasma lemma is a static layer. Second, it doesn't explain the phenomena like active transport. Now, Robertson model. This model is also known as unit membrane model, and it was proposed by J. David Robertson in 1995 AD. He studied ultrastructure of a number of biomembranes of the cell, that is plasma membrane, nuclear membrane, mitochondrial membrane, And what he found? He found that all these membranes are similar in their ultrastructure except in their thickness. He coined the term unit membrane and stated that each biomembrane is formed of one or more units of membrane. According to Robertson model, biomembrane is a lipoproteinous and trilamellar. Protein molecules here are extended, that is beta protein or fibrous, and they are not globular. Here, each phospholipid is a bipolar molecule with a hydrophilic head of glycerol and hydrophobic tail of two fatty acid chains. Head of phospholipid molecules are directed in opposite direction, while tails of two layers face each other. Average thickness of this membrane is 75 to 100 angstrom, in which each, that is inner and outer protein layer, is 20 to 30 angstrom, while phospholipid bilayer is 35 to 40 angstrom. So, both Danieli and Devson model and Robertson model of unit membrane are collectively called lamellar model. It has same drawbacks as of sandwich model hypothesis. The same, that it does not explain the functional specificity and variability in the biomembrane, and also doesn't explain the phenomena like active transport. Now, fluid mosaic model. It is a model that describes the composition of plasma membrane and how phospholipid, proteins and carbohydrates freely move within it. This model was proposed by S. Jonathan Singer and Garth Nicholson in 1972. Actually, it is based on the information gathered from chemical analysis and biophysical properties of plasma membrane, and it is one of the most accepted models. According to this model, plasma membrane is formed of protein iceberg in sea of lipids. This model is characterized by plasma membrane. According to this model, plasma membrane is lipoproteinous and trilamellar. Lipid bilayer is fluidy in nature, in which phospholipid molecule two types of movements, that is transition movements and flip-flop movements. Here, see, if this is a molecule, it changes position within the same layer. That means if this comes here, then it is a transitional movement. Then second movement is flip-flop movement. Molecules of two layers can be interchanged. So if this model comes here, if this molecule comes here and this comes here, so they are interchanging between two layers. So this is flip-flop movement. Now, in this model, proteins are globular and they are of two types: extrinsic or peripheral and intrinsic or integral. So here you can see first in this model, some intrinsic proteins are so large size that they project on both the sides of surface of phospholipid bilayer, and they are called tunnel or transport protein or transmembrane protein. This extended throughout the phospholipid bilayer as a single helix, that is a glycophorin. They act as a charge channel for the passage of water-soluble material along the concentration gradient. So, this is a intrinsic or integral protein. Here you can see this is also integral protein. Now, peripheral protein. Here you can see these are peripheral protein. Here there are this is also protein. There is one more thing, glycoprotein. This is carbohydrate chain. When there is a chain of carbohydrate molecule attached with the protein, then it becomes glycoprotein. Now here in this model, each phospholipid molecule, see this is each phospholipid molecule is a polar molecule and it is two specific ends. The same one, hydrophilic head of glycerol and hydrophobic tail of two fatty acid chain, like previous model. So again, this is a amphipathic molecule, the same as that of previous models. Heads of phospholipid molecules of two layers are directed in opposite direction here, while tails of two layers face each other here. And this arrangement forms a water resistant barrier. Now carbohydrate molecule, especially oligosaccharides, they are associated with outer surface of plasma membrane. These either form glycolipids when they are associated with phospholipid molecule. Here I will show. This is there are also glycoprotein. These are glycoprotein. Glycoprotein is formed when protein molecules get associated with lipid molecule. So it is a glycoprotein, means when carbohydrate molecule is associated with protein molecule, then it forms glycoprotein. There are also sterol molecule, especially of cholesterol. Here you can see they are present in between the phospholipids. These sterols are more rigid and provide stability to animal membranes. So due to this quasi-fluid state of this model, biomembranes can undergo dynamic changes and help in cell growth, formation of intercellular junctions, secretions, endocytosis, cell division, etc. So plasma membrane has two distinct surfaces, that is one cytoplasmic surface and exoplasmic surface, because different types of phospholipids are found in two layers, different types of protein in two protein monolayer, and glycoprotein and glycolipids are found only on exoplasmic surface. So this is all about fluid mosaic model of plasma membrane. So today we discussed two models of plasma membrane, that is a lamellar model and fluid mosaic model. Lamellar model comprises of two models, that is a Danieli and Davison model and Robertson model. Danieli and Davison model is also known as sandwich model, means protein lipid protein sandwich, and it is proposed by J. Danieli and H. Davison in 1935 AD. Then Robertson model, which is also known as unit membrane model, it was proposed by J. David and Robertson in 1995. And then fluid mosaic model of plasma membrane, which describes the composition of plasma membrane and how phospholipids, proteins and carbohydrates freely move within it. Fluid mosaic model was proposed by Singer and Nicholson in 1972. This is a fluid mosaic model where I show two types of protein, that is extrinsic or intrinsic. See, this is integral protein. This is also integral protein, and this is a peripheral protein. Then glycolipid, when carbohydrate is attached with lipid, then it becomes glycolipid. Then when carbohydrate is associated with protein, then it becomes glycoprotein. And this is a tri-layer structure where it is a protein, this is protein, and between this there is a phospholipid bilayer. This is protein here, this is protein, and this is a phospholipid bilayer. So it is fluid mosaic model of plasma membrane. Thank you.